So today we'll start a new chapter, anti-tubercular drugs. So first of all, we'll learn about what is this tuberculosis. Tuberculosis is a chronic granulomatous disease and a major health problem. About one third of the world population is infected with the Mycobacterium tuberculosis. So what is this tuberculosis? This tuberculosis is a chronic granulomatous disease and a major health problem. About one third of the world population is infected with this Mycobacterium tuberculosis. So, this tuberculosis is a type of disease that is mainly infecting about one third of the world population, and the main that is the name of the a particular bacteria that mainly infect the person is mainly called your Mycobacteria tuberculosis. Now, what are the classifications of the drugs? That is your first line drug and the second line drugs. So now we have to learn about what are the first line drugs. The first line drugs are, you can see that isoniazid, ethambutol, rifampicin, streptomycin, pyrazinamide. So these first line drugs, these are called the first classification, and the drug that will fall under these first line drugs, these are isoniazid, ethambutol, rifampicin, streptomycin and pyrazinamide. Now we have to come to the second line drugs. So what are the second line drugs? The second line drugs are thiacetazone, newer drugs, para-aminosalicylic acid, ciprofloxacin, okay, then ethionamide, ofloxacin, clarithromycin, cyclosadine, azithromycin, kanamycin, rifabutin, amikacin, capromycin. So these are called the second line drugs. So you have to learn carefully the classifications, okay, that is what are the drug that are present under your first line drug, and what are the drug that is present under your second line drugs. Now we will come to the mechanism of action of the INH. So what is this INH? This INH means isoniazid. This INH means isoniazid. That means the short form of the isoniazid, we can call it as an INH. See here, you can see this is called your isoniazid, okay, and the short form, in short, we can write this isoniazid as a INH. Now what is the mechanism of action of this INH? We have to learn this, okay. So you know that what is cell wall, and when we have learned the chapter your beta-lactam antibiotics, in that chapter we have learned thoroughly about the structure of the bacterial cell wall, and in that structure we have also learned about the peptidoglycan layer, how this cell wall is made up of, what are the ingredients present in the cell wall, and what are the ingredient mainly the cell wall is made up of, that is your NAM and NAG, that is your N-acetyl glucosamine and N-acetyl your muramic acid. Here I have already downloaded the picture, you can see from here. See, I think all of you can remember this picture, okay. This picture is the structure of a cell wall. How this cell wall is made up of? The cell wall is made up of mainly this NAM and NAG. This is L-alanine, D-glucosamine, L-lysine, D-alanine, D-alanine. So this is the structure of the cell wall, and here you can see NAM and NAG. This NAM means N-acetyl muramic acid and NAG means N-acetyl glucosamine. So there have been there is a cross linkage, okay. So this is a cross linkage, and see this D means D-alanine. So this is a cross linkage, okay. This is a type of cross linkage that mainly formed in the cell wall, and this bond is mainly called this bond is mainly called your peptide bond, and this peptide bond is mainly prepared by your transpeptidase enzyme, okay. So this is the structure of the cell wall. Now one of the important component of the cell wall is your mycolic acid. One of the important component of the cell wall is your mycolic acid. So what is this mycolic acid? This mycolic acid is a particular fatty acid component of the mycobacterium cell wall. So what is this mycolic acid? This mycolic acid is a unique fatty acid component of the mycobacterium cell wall. That means we can clearly tell that this mycolic acid is an important fatty acid component, and it is an important component of the cell wall. In the previous class, in the class of the lactam drugs, I have told you that the cell wall is made up of your particular structure. Just now I showed you the picture, and in combination this today we are learning that this mycolic acid is also a component of the mycobacterium cell wall. This mycolic acid is also a component of the mycobacterium cell wall. So this cell wall is a kind of barrier. This cell wall is a kind of barrier by which a bacteria can prevent itself from any kind of damage. Okay, for example, I am sitting inside the room. Okay, I am sitting inside the room. If an enemy will come from the outside, that enemy have to invade my room. Then only this enemy can damage me. Okay, but if as I am present inside the room, a room is there, and you imagine this room is a cell wall. Okay, this is a cell wall. That is that enemy have to invade. The enemy had to invade inside the room by permeating through the room. Then only this enemy can damage me. Like that, in the case of bacteria also, this cell wall is a kind of barrier. This cell wall is a kind of barrier by which this bacteria can prevent themselves from any kind of damages. Okay, and this bacterium cell wall is made up of various kind of fatty acid is there, various your peptide bond is there, and then your D-alanine, L-alanine linkages are there. And actually this is a kind of cross linkages that is mainly present inside the cell wall. And these cross linkages and this mycolic acid and these cross linkages, all of this mainly gives a stability and integrity to the cell wall. Okay, so so it is very clear that if this cross linkages and if this mycolic acid get damaged, then the cell wall will get damaged. In the beta lactam class, I have already told you that if the bacterium cell wall will get damaged, then what will happen? There will be formations of the pores over the cell wall, and what will happen? Outside water will get inside the cell, and what will happen? The cell will burst. Okay, and the bacterial lysis will occur. So that means this mycolic acid, this N-acetyl muramic acid, this N-acetyl glucosamine, this D-alanine linkages and the N-alanine these linkages, these are very important for the particular stability and the integrity of the bacterium cell wall. Okay, so in that case, various kind of drugs has been synthesized. So to break this cell wall, okay, and to kill the bacteria. Okay, because it has been observed that if any drug can break the bonding inside the cell wall, then what will happen? The cell wall will break, and some pores will create over the cell wall, and for that reason the outside water will able to get inside the cell, and what will happen? The cell will burst. Okay, বাইরের জল যদি তোমার ভিতরের মধ্যে ঢুকে যায় তাহলে তোমার সেলটা আস্তে আস্তে বড় হয়ে যাবে, আস্তে আস্তে বাস্ট হয়ে যাবে. This is one kind of lysis. Okay, that is a bactericidal case. So depending upon this mechanism, various kind of drugs have been synthesized which will mainly work depending upon this mechanism. And today we'll learn about this isoniazid, and this isoniazid is also a kind of anti-tubercular drug, and this drug mainly block this mycolic acid synthesis. And if this mycolic acid synthesis can be blocked, then what will happen? The bacterial cell wall will lose its integrity and stability, and what will happen? The bacterial cell wall will get damaged. So this is the important mechanism of action of this isoniazid, and you go to the next video, I will tell you in details about this isoniazid.